So, Shanique Alabi, you're a scientist at Monte Rosa Therapeutics, which is in Boston somewhere, in the Boston area.
Yes, in Seaport.
Right. So you started your academic career at Cornell when you did your BS in chemistry and chemical biology. There's an interesting aspect here that I'd like to dig into afterwards. But anyway, we'll get there.
Okay.
So you went on to study pharmacology at Icahn School of Medicine, and then you started your PhD work with Cruz at Yale. And after that, you went on to be a Yale Canon Fellow and a Gilliam Fellow, and now a Hughes Medical Institute. And then you joined Monte Rosa. Is that correct?
So the last few were actually like during my PhD.
Oh, okay.
So, what was the first one you said? The Canaan Fellow?
Yes.
So Yale's like PhD program has like a lot of different additional programs you can add on to your PhD almost, which is like one of the reasons I picked it. So the Canaan Fellowship is actually something that I did like I think my fourth year of my PhD. So it's during. So the HHMI Fellowship is, just a predoctoral fellowship that I had. So that's also during my PhD. And then the last one was
the Gilliam Fellow at the
yes.
Howard Hughes Medical Institute.
Yes.
So are these things adding to your project? Are they sort of parallel projects or added projects to your core PhD program? Is this the way it works?
I did all of this during my PhD. So the Gilliam Fellowship is a fellowship that I applied to that kind of funded my predoctoral work.
Okay, I understand.
And they had like components where we'd go down there once a year, and they'd have like workshops for scientists and all the above. But yeah, it was all during my PhD.
Well, of course, when you're at Yale, there must be options there, right? And there's things happening. Anyway, it obviously sounds like an interesting path already, and we're gonna just get there and hopefully get some more details from you, and that's the way we really start usually the conversation, which is kind of trying to look back and understand how you got where you are. It's as simple as that. So let's speak about you a little bit. So if you look back, how did you even start being interested in science? Let's start from there.
Let's see. I guess like I have like my personal statement answer is what I call it, right? I think the first time I'd say I gained an interest in science, I could place a couple places. I can either place it maybe in middle school and high school when I had really great like science teachers, or I can even place it before that when I was in Ghana and, I kind of had an interest in what makes a drug essentially. Because I had, essentially my grandfather has a pharmacy in Ghana and, it's kind of like our family business and it's still running up to this day. And as a kid, so I spent like a little bit of my childhood in Ghana, about 10 years. And as a kid, like on the weekends, sometimes I'd go there and help out. And that's like, I feel like my personal statement, that's why I call it my personal statement answer almost, where like my curiosity for what is a drug, what makes a drug, how can I give you this pill? And all of a sudden you feel well. And that was like almost a kid's curiosity, I guess, because I just knew this is something we sold. So yeah, that kind of, those are kind of my two answers as to what got me into science. Yeah.
Which one do you prefer?
Which one do I prefer? Which one sounds better?
Well, the family origin sounds better, Yeah, you like science at school. That's something that's very common for many people. Not many had experience with, some sort of pharmacy business in the family. That's actually fascinating. Anyway, and you also mentioned already something which is quite unique, right? You grew up between the US and Ghana. You were actually born in the US, weren't you?
Yeah, I was. My dad came here for college, and then like him and my mom actually like were dating in Ghana before. And then my mom came here after he was done with college, and they were kind of setting up their lives and just like struggling with having kids and setting up their lives. So my sister and I actually went to Ghana when I was 2 and my sister was like a kid, and we stayed there in my family house with my aunt and my grandparents and cousins and such. Yeah.
While your parents were trying to figure it out.
Exactly. Okay. The immigrant story. Yeah.
Well, of course, it's very understandable. So you're not properly an expert, but you still live this sort of multicultural international reality, right?
Yeah, it was great because like, and I can now speak the language. You know, I know my family a lot better than I would have if I stayed here. Yeah.
And you spent a fairly long time. You mentioned, did I understand correctly, 10 years?
Yeah, from 2 to 12. Yeah.
Yeah, these are the years that define you in many ways.
Very much so. Yeah.
How, you know, do you think this gives you an edge or something? Make you unique or different in many ways? If you look at your normal peers.
Absolutely. I think it gives me an interesting perspective. I feel like I always see things in many ways because of it. I feel like, yeah, my life here and my life there and being able to kind of exist in both worlds, which are pretty different. So,
I bet. What is, tell me something about your childhood in Ghana. I mean, do you have a good memory? Were they easy years? Were they fun years?
Yeah, I remember it really fondly. There were years where I played a lot of tennis. I was like, I loved school. I loved going to school. I spent a lot of time with my cousins, so it was like a pretty lively childhood with lots of like people around. Yeah, I remember it very fondly.
So, how's the education down there? Is it strong? Because your records are actually pretty good. I mean, you end up in Yale, and so there must be something in there.
Yes. So, as with everything, right? The resources that you have give you,
of course,
the level of education that you have. So, I think while maybe if I were to have stayed here with my parents, it would have been a little bit tougher. Back there, I was able to go to. Actually, it wasn't technically an international school, but Rich Church School. It's like it was a private school, and it was a very good school. And the education system, I will say, like math, for instance, is really spectacular. I don't think I learned new math for a couple years when I came back here. So it was like definitely a strong educational background. Yeah, I mean, I have always loved school, and when I came here, I mean, I liked science fairly early on. So when I came here, I was in public school at this point because private school in Ghana is affordable. Private school here, not so much.
Yeah.
But Northern Virginia is known to have a really great like public school education system, essentially. And Prince William County has like a number of different schools that specialize in different things. So for high school, so after like going to like my local middle school, in high school, I went to kind of the biotechnology school, so Osborne Park High School, which had a focus on science. And there I kind of, I don't know, I worked hard. I picked the AP classes. I picked the honors classes. I was with the group of students who were like, on that track. So, I mean, in some ways it was normal because like, that's what all my peers were doing. So, in other ways, like it was kind of, I like school and I like science and I applied myself in that way. So, yeah.
And that's pretty cool. That's where you started looking into this sort of interfaces between chemistry and biology, right? If I understood correctly, you did some homology modeling, vitro screening, so started to looking into how things look like, how a molecule works and how, what shape it has and stuff like that. That's fascinating. It resonated with me because I did a similar thing when I was as young as you were back then. But yeah, that was.
I actually did that between Cornell and before my PhD. So that was actually a little later in my life.
Okay, that was later.
Yeah. In high school, actually, didn't do much research. I didn't do research at all in high school, but I definitely loved science. I always loved chemistry and biology. Those were like my two favorite subjects. And actually picking a major at Cornell was pretty hard because I liked both.
Yeah. I'm glad you're going there because it comes across quite clearly, right? You almost seemed indecisive between the two. So you love either, and then you kind of did a bit of one, a bit of another. You ended up doing this sort of maybe chemical biology type of work in many ways, or even these pharmaceutical sciences type of things. They tend to fit in the middle, right? Is this just a compromise for you because you couldn't really pick, or how do you see it?
Oh, I think luckily the field of chemical biology existed before I ever, ever came into the field.
It's fair. Yeah.
So I didn't have to invent it, but I think that's just naturally where my interests fit. Like, I don't know the thought of like how small molecules interact with the body it's fascinating to me. So yeah.
And did you ever thought when you were going there to your early years and experiencing, the pharmacy business in your family, were you ever thinking, oh yeah, I'm finally starting to understand how these things work?
Oh, while I was during my education,
yes.
Not as directly, which this is why I call it like my personal statement answer, But
I appreciate your honesty here. But you could have lied. It would have been nice anyway.
I need to learn. I need to learn how to craft my story a little better.
This is part of the process, so maybe next time.
my next interview, I'll have it down packed.
Yeah, absolutely. It is interesting, and just trying to be serious. I think it's fair to say that the most exciting science is happening at the interface between disciplines, right? And so chemical biology is really well positioned. And I believe, I don't know what's your opinion, but I believe that looking at biology problems with a chemical lens is something new, relatively speaking, and possibly gives you an edge. Would you agree with that?
I will say that it allows you to look at things maybe a little bit more granularly than like a general biologist might. I think it definitely, overall, I think having more perspectives on an issue or problem is never going to be a bad thing. So I think, I wouldn't say that chemical biologists have the best insight, but I think we bring really good insight to biology for sure.
Sure.
And I think there are a lot of biological, discoveries that kind of hinge on, a small molecule inhibitor was discovered to do this, and now we understand this pathway, So it definitely, it doesn't hurt. Yeah.
Yeah, I am a chemist by background, so I tend to look at things, from that perspective. And for me, biology is just super complex chemistry, but it's a sequence of reactions.
That is true. Yeah.
And so pathways just use a lot of catalysts, which are, just proteins, really. I know I'm oversimplifying a little bit, but if you speak to a biologist, my wife, for example, they don't see things this way. But they have a more holistic view very often, which is also very important. So kind of finding your position there is key, isn't it?
Absolutely. Yes.
So have you have any regret about, your choices in your education?
I would say no.
Okay.
I don't think so. Sometimes I wish I picked a biology major instead of a chemistry major.
Oh, interesting.
But yeah, no, overall, I don't have any regrets.
Why is that? Why do you sometimes think that?
Because I feel like, regardless, I ended up taking a lot of biology classes, I will say. but I feel like the complexity of biology sometimes is something that I don't know if I have a broad understanding of. Like, there's some fields in biology where I'm like immunology, for instance, where I'm like, yes, CD4 positive cells. And when you're out there, I have no idea, So I feel like for some things like that, granted, I feel like they can be learned, right? You can learn them when you need them. Exactly. But sometimes, having that broad understanding of things, or that broad kind of like baseline of knowledge is helpful in biology as well.
Yes.
Yeah.
Yes.
And maybe it might have been more important than me learning PChem, for instance. Yeah.
I attended your presentation when you got your Talented 12 award. Congratulations for that. By the way, I think you are the youngest in the 2021 class. Am I right?
Yes, in the 2021 class, yes. I think I am. Yeah.
Yeah.
Not in all the other classes, but 2021, yes.
Well, that's good. That's something to be proud of. So congratulations for that.
Thank you.
Something that caught my attention was the way you discussed about Cruz once you were choosing your PhD program, right? Or trying to understand what you wanted to do. So I go back there, but that makes me think about the role of certain people during your education. I'm sure you have had a few mentors, and probably Cruz is one of those. And I know for many of the people we interview, your mentors tend to play a key role, right, in the development. for some people more, for some people less. But would you say you had many, or at least a few very special ones during your educational career?
Oh, absolutely. Yes, solid yes. I had several. Some I'm probably forgetting. Yes.
Okay. Do you want to speak about some of them?
Let's see. The first advisor I kind of had at Cornell. So his name is Manuel Irigoyen. He was a senior research scientist at Cornell in the plant sciences department. And I worked very closely with him, and he was really important to, like, one, teaching me about, like, what is research? How do you ask a question? How do you craft an experiment to answer that question? And he also told me about the PrEP program, which is a post-bac program. And that's why I spent a year at Mount Sinai doing research. Because after I left Cornell, I actually wasn't too sure whether I wanted to do research or not. I had some research experience, but not enough to, I feel like, warrant a decision to say, yes, this will be my life. So him like telling me about this program was really great. And there, I had, oh my gosh, several mentors that set me up to where I am today. I'll start off with my PI there, Avner Schlesinger, and he was the guy I kind of did homology modeling and virtual screening under. I didn't have any computational experience whatsoever, but I was like, okay, I want to do something in a year. I want to go to grad school in a year, I think. So I want to be able to be productive, and I want to be able to still be in the interface of biology and chemistry. So it was just like a perfect fit to do homology modeling and virtual screening. And he took me on, and I had to learn fast. And it was like challenging, but it was good. And he was just a great guy and a great mentor overall. And there, like in the program as well, we had other PIs and other advisors there too, who really helped us craft a graduate school application, taught us what it means to interview at a school, what's important, what's not, how to describe your research. Jeannie Hirsch is one of them, and Terry Krolrich is one of them. And they were phenomenal. Like, I have goosebumps thinking about them right now. They were phenomenal. So I'm really, really thankful for them. And then at Yale, oh man, working with Craig has been an amazing experience. Working with Craig is an amazing experience. The guy's just brilliant, and he has so many ideas, and I learned so much from him as well. Yeah.
Well, you gave an interesting answer. It's quite unique. Obviously, you showed this sort of emotional attachment to some of these people, which is amazing, right? That's the story of the people, the person you are. So it's really good. But it's also very, very easy, right, to be inspired by these big professors. You know, they're usually very charismatic, and
Craig is charismatic.
Yeah, and so I can see why you know you're fascinated by these people, and then they become your mentors and they inspire your choices, and that's good. But you know, you mentioning, you know, our senior researchers working with you and helping you out in your first steps in the lab, and giving this sort of pragmatic support on how to run experiments, how to interpret the results. I mean, there's a lot of these people, and very often they are even postdocs or even PhD students, maybe at the end of their career, and they play such important roles for people just starting in the studies. And I don't know, sometimes I think they are forgotten, but these are really important for most of us, right?
That is very true. They're the ones standing literally right next to us as we figure out what it means to pipette. So yeah, absolutely.
Exactly.
Yeah.
Exactly.
Yeah.
And if you don't learn that one, then you can't really win any award afterwards.
No, you can't.
Yeah. Yeah. So let's get to Craig Cruz then. You said that once he presented his research programs, they were pitching to the potential student candidates, and you immediately decided that was what you wanted to do. Why is that? What was so captivating there?
Sure. So I kind of, as you're applying to schools, you're told, or I had such good advisors that I was told to kind of look at the school, look at the different faculty that are there. You have to find 5 people at least who you would want to work with. If you can't, don't apply to that school. That's kind of the advice I was given. And I had actually seen Craig's work, and I'd seen a couple of other people. So Dave Spiegel, for instance, and they were doing heterobifunctional things. So, you read about inhibitors, you read about RNAi, CRISPR, there's just stopping function. But just the idea of bringing 2 things together, I thought was fascinating. That was one of the strong reasons I applied to Yale.
So he was shortlisted already for you, even before meeting him.
Yes, but actually I kind of had forgotten about, so I did my first rotation in like crystallography, and then I did like some other chemical biology stuff with, he was shortlisted, but like I'd forgotten that I had that interest after like my first rotation. And then I saw that presentation. I was like, oh my gosh, yes, how did I forget about looking at this page? Yeah. And he's like a really good speaker. He walks you through what he's thinking, and it was just like, amazing talk. So, must work there.
So was this the sort of different way of thinking, as you say, bringing different concepts together feels like a bit more innovative?
Yes, it was definitely innovative in a way that I hadn't seen. Like I hadn't seen anything like it, and I thought it was super cool. And I still think it's super cool after years of being in the field. Yeah.
So then you landed in his lab and you started your work. Shall we go and speak a little bit about what you did there then?
Sure. Yeah. So there, I worked on a number of projects. Earlier on, I was like kind of learning the ropes, working with older graduate students who were all great. Jamila, Dan, Ashton, they were all amazing scientists. And then I guess the project that took off for me was looking at degradation of BRAF, which is a mutant protein. Well, it's a proto-oncogene, a protein that can become mutated in the cell and cause a number of diseases. More like it's mostly known for melanoma, but yeah. So that became my main project, looking at the effects of degradation of BRAF mutants.
Okay, so was reading a little bit of your work, and I obviously, I wasn't aware of these sort of approaches, and I found it really interesting. It's really out of the box thinking. So my understanding, and I'm trying to oversimplify, but then you're explaining as an expert better than I can. The idea here is using or influencing natural processes that look at degrading proteins after they have done whatever biological role they have, to actually influence or regulate, expression or overexpression or underexpression of proteins when they don't really work the way we want, like during diseases. It feels like a completely out of the box therapeutic approach to me. Am I more or less there in my understanding?
No, yeah, you're exactly there. I think it's in some ways it's out of the box, in some ways it's not. So I mean, in terms of like, proteins going awry, right? Like people try to stop their functions in many ways, and get rid of their function completely in many ways. But the way this does it is really cool. Yeah, like you said, proteins are, the macromolecules in the cell. They're kind of really well regulated, right? They're expressed at a certain time and they live, and then they're degraded like us. They expire at some point. And I know, so people usually you try to inhibit the function of a protein usually by mimicking something that it normally binds and actually like blocking its function, or using RNAi to get rid of the protein before it's even expressed. But being able to kind of take advantage of the cell's natural process of getting rid of proteins is really cool, right? Because it's just a different way of getting to a thing that people have been getting at in different ways. Yeah.
Can you explain to the audience how these degradation processes normally work and how you exploit it actually for therapeutic approaches?
Sure. So proteins are recognized by this degradation machinery in the cell. It's called the ubiquitin proteasome system. So each protein has a cognate E3 ligase, and that's the name of another protein that kind of recognizes this other protein, and then it marks it with a post-translational modification. So it's like almost like a tag for degradation via the proteasome, which is like the cell's recycling can, is what I call it, because the amino acids can go in and be used.
So the proteasome is able to recognize the tag, and so every protein which is tagged is a victim pretty much. Sooner or later, it encounters the proteasome, it gets degraded.
Exactly. Yeah. There's a specific chain that is recognized by the proteasome and is degraded. So the way targeted protein degradation takes advantage of it is that you take an E3 ligase and you use a binder, so a binder to this E3 ligase, and have a binder for PROTAC specifically to your protein of interest, and you kind of force them to interact. So this can be a protein that would normally interact with this E3 ligase, but by forcing this interaction, you allow for this protein to then be marked and be recognized and degraded. So you were
So how does it work in the cell? Does the kinase at some point encounter, this is a statistical process, at some point the kinase is going to encounter the protein because there's a lot of it, and then it gets tagged and then degraded? Or is there any other mechanism that attracts the kinase to the target protein?
Is this for regularly or with
normally? Yeah, normally.
Normally, yeah. There's several ways which proteins. So it can either be it's usually called a degron. There are lots of different types of degrons, and E3 ligases recognize degrons in different ways. The common one is like an N-terminal degron, which this protein is kind of evolutionary designed to recognize, and then it's degraded. So normally, like a regular protein has a specific E3 ligase that comes in and does this function. Yeah.
Okay. And then you mentioned the fact that you're actually affecting this process by artificially attracting the ligase to the target protein somehow. And you mentioned the PROTAC. Well, now is the time to explain what that is then.
I did. I just dropped it in there.
Yeah.
So PROTAC, so this was what was kind of the thing that was exciting to me, right, that Craig talked about. It stands for Proteolysis Targeting Chimera, which is a mouthful. Proteolysis, meaning that it breaks down proteins. Targeting, meaning that it's, targeting chimera. I'm going to explain those together. Chimera meaning something with, two heads, and targeting these two things together. Yeah. So PROTACs.
So the idea, I love this because the idea is so simple and elegant, right? You develop a molecule which has affinity for both the target protein and the ligase, so that they can actually go closer together, and then the ligase is in the perfect position to attack ubiquitin and then basically get rid of that protein.
Yes.
It's, as I said at the beginning, I find it simple and elegant. Did you get this feeling at the very beginning when Craig was describing it?
Yeah, absolutely. I thought it was fascinating. Yeah, super cool.
So you apply this concept to the BRAF, which is an overexpressed protein in some cancers, am I right?
So it can be overexpressed. So BRAF it's a commonly mutated protein in cancer specifically. So it's a kinase, which is a protein that phosphorylates other proteins. And it's known to be mutated in 8% of all cancers. So these are usually different point mutations. So, alanine to valine type of mutations that cause BRAF to increase. It essentially increases the pathway, which causes increase in proliferation, differentiation, and survival essentially of cancers. Yeah.
And are there any existing, more traditional, if you wish, therapeutic approaches to this type of problem?
Exactly, yeah. Which is kind of embedded in PROTACs, right? Having something to be able to engage this, right? So there are, but only for one mutation. So which is it's actually the most common one, so that's good. But there are also hundreds of others that are untargeted. So there are 3 FDA-approved BRAF V600E inhibitors that are actually in the clinic. And they work well for some time, but unfortunately with kinase inhibitors in the clinic, they usually work only for about a few months. And for these like vemurafenib, it's usually between 6 to 9 months in which they work. Yeah.
Why is that? Does the protein keep mutating?
Yes, it's a number of reasons. There are so many ways in which like the cell kind of tries to overcompensate for the fact that this protein is gone. It can either overexpress another allele of BRAF, additional mutations that kind of stop the small molecule from binding, upregulations of things upstream and downstream of BRAF. Yeah, there are a lot of mechanisms that kind of compensate for it. Yeah.
Hence the complexity of biology, right?
Yes,
so
exactly. Exactly. That's what your wife would say.
Yes, exactly. And then we probably don't even understand everything, which is the fascinating part of it. And so how do you design these molecules? So did you take any of these existing inhibitors as a sort of template to at least for binding the target protein? Is that the way it works?
Exactly. So we took. So I worked very closely with a chemist for this. So I'm not a synthetic chemist at all.
Okay.
So Saul and I worked very closely together. So we picked vemurafenib, which is an FDA-approved drug, and it's one of these 3 inhibitors that I just discussed, and linked it to an E3 ligase E3 ligase binder, which was designed in the lab. it's a VHL ligand. And we tried a number of different linkers between them to get the perfect PROTAC, and that's what I've published.
What kind of, so how did you go along with designing this sort of the linkers or the overall structure? Is it like a combinatorial approach, or do you look at synthetic feasibility? How does it look from the design of the experiment's perspective?
All the above. Okay. I mean, the linker composition is actually like, I feel like it's the tougher part of PROTAC development, right? Because you can imagine it could be very flexible, it could be very rigid. So essentially, you kind of want to get a sense of, how deep are these pockets, how much room do you have to like play with in order to have the optimal linker length. And with that knowledge, we kind of tried a number of different linker compositions until we got the one that worked the way we wanted.
Okay. Yeah. So you were designing and testing and then improving the design.
Exactly.
Is there any sort of computational approach where you're trying to simulate interaction and things like that to kind of learn from it?
Back then, no, but now actually that has really taken off in the PROTAC field, as you could imagine. Yeah, that's probably a grant out there. Exactly what you just said. Yes, yeah.
Yeah, it's not my money, but I would. Yeah, I've loved the idea, but I can't afford it. This is fascinating. So, how did it work?
In terms of?
Yeah, the results. Where your PROTAC working? What was the ultimate result? Were you able to inhibit and then degrade the protein effectively? Do you have any sort of in vivo results supporting that this is potentially a good way to go?
Yeah, so it worked well. It not only inhibited BRAF V600E, which is, what the inhibitor itself was already kind of designed to inhibit. We found that it actually was able to bind and degrade BRAF V600E and all the other classes of BRAF mutations that have been kind of described, including also some truncations actually that occur after people have been given vemurafenib that stops the effectiveness, which was super cool. And even better yet, we found that it did not touch the wild type allele. So unmutated BRAF was kind of left alone. So it was really great. And we were able to test, one, did it have an effect on cell growth? And we found that, yes, it had a really good effect on cell growth. It was able to inhibit cell growth better than the inhibitor itself, which was great. And also we were able to prove that, yes, it does work in vivo, that if we gave a mouse that had a xenograft, we were able to see a decrease in tumor growth.
Well, this is fascinating.
Yeah.
And do we know why they don't have the same sort of long-term issues as the competitive inhibitors? Because at the end of the day, they still interact with the protein. Why do we understand that?
So I mean, the field is a super. It's nascent field, like stages. So people are now starting to kind of study what resistance to PROTACs will look like, because they're not special. Resistance will form to them.
Of course. Yeah.
And we're finding that actually what we're finding is that resistance. I mean, so the drug I just described is not in clinic, so I don't have exactly an answer for you. But what the literature has been showing is that actually resistance can form to PROTACs, and what happens is that actually whereas with inhibitors you see an effect on, say, BRAF itself, we're actually seeing an effect on the E3 ligase function. So that's where actually I think there are a few papers now that look at resistance mechanisms in a number of ways, and it's actually been consistently at the E3 function of the PROTAC versus the target protein, which is fascinating. Yeah. So
how long has the approach been out for? So is it something very new? Or it feels like there's a lot to still be understood.
It's technically been 20 years, so it's a little older.
That's what I thought. Yeah.
But it's as something in the clinic, very new. The first PROTAC went into patients maybe 3 years ago. I'm not sure exactly, but very, very recently. But it started off. So
there's already clinical use of some of these.
Yes, products. Yeah. So Craig started a company, Arvenis, that is in clinic with PROTACs now, and there are a number of other companies that are doing that as well. But so essentially, in some ways, the field is a little older, but it started off kind of more of a chemical biology trick. The first PROTAC was large, and it was given to lysates, we couldn't even give it to a full intact cell. It was peptidic based. It was very, very more of a kind of something you do in the lab and die there. And after it became more small molecule based, the field kind of took off. Yeah.
Yeah, and that's the beauty, right? Because you know you can use small molecules, so actual traditional synthetic chemistry, and while still looking at very complex biological problems, and just being a chemist and being a biologist at the same time. Now, this is what I really find interesting, and some great results and out of the box thinking are coming from this perspective, which is really good. So do you know if your work, because obviously you finished your PhD and you moved on, but do you know if your own work is having like a future? Or is this still something that the team is working on and developing further?
I'm not entirely sure. Yeah, I'm not sure about that.
Fair.
Yeah.
Fair.
I think there's,
Fair.
Definitely maybe something percolating there, but I'm not sure. Yeah.
I'm assuming that Craig is working on a number of different targets.
Yes,
and a number of
Craig has a lot of
different
ideas. Yeah, this is old school, old news for him. He's like moving on to much newer, cooler things. Yeah.
I will now move to to what you're doing now after your PhD. You move on to Monte Rosa, and I'm assuming that your work is probably not the same thing, but it is based on similar concepts, right?
Yeah. So I now work on what we call molecular glues, so molecular glue degraders specifically, and they're actually technically under the kind of same umbrella as, essentially things that kind of hijack the ubiquitin proteasome system. But molecular glues are actually technically an older class. We just didn't know it yet. So, technically molecular glues have been approved. So thalidomide was the first molecular glue degrader, that was approved.
We didn't know it was working that way.
No, we didn't. We didn't know that till 2010. Yeah, but essentially the way it works is that it binds. So the way molecular glues in general work is that they bind an E3 ligase, specifically for thalidomide, cereblon, and kind of reshape the surface of it and allow it to recruit what we call a neosubstrate. And the thalidomide, one of the things that it recruits is SAL4, and then cause the ubiquitination of it and cause its degradation.
Oh.
So it was given back then, right, to pregnant women, and unfortunately it caused a lot of birth defects. And it really revolutionized how the FDA really deals with drug approvals, which is a good part of what came out of all of this. But essentially, there was still a lot of interest, right, in thalidomide, and primarily because of its immunomodulatory effects. So that's kind of why it was restudied again. Right now it's approved for multiple myeloma. I think it was approved for leprosy or host versus graft disease. It's approved for a number of things right now.
Oh, I didn't even know. I thought it was completely forgotten. So it's still used actually.
Yes, it is. Yeah.
You just need to ensure you don't give it to pregnant women.
Yes, exactly. Now we understand how it works. And it's actually, I think it's really highly regulated, but it's still used. Yeah.
Of course. So the idea is still basically stimulated the interaction between macromolecules in the cell somehow, right?
Yes.
Through a small molecule of some kind.
Yes. Molecular glues, they don't necessarily have that modular effects where you have an inhibitor, E3 ligase binder, a linker. They're a lot smaller, and they just kind of modulate the surface and allow recruitment of proteins. So in a lot of ways, I get to use the kind of the knowledge base I had working on PROTACs, but this is completely different. It's a different beast completely. It's very interesting too. Yeah.
Of course. It sounds very interesting. What do you focus on? Is it more on the design of new molecules, or do you study the mechanisms? What is your main area of focus?
The mechanistic studies, yes. Okay. I'm very interested in ternary complex formation, and I still do that up to today. Yeah.
Okay. Is Monte Rosa, does the company have an amount of compounds that are already of clinical interest? Is there any more promising molecule among the ones you work with or the company works with, or maybe something which is already commercial from the company?
So we're a much newer company. We're in Boston, maybe 2 years old, and Basel a little longer. Mm-hmm. But essentially, we So I should probably rewind a little bit. Yeah. So molecular glues in general, so beyond thalidomide, right? We've come to understand a lot about what allows for a protein to be recognized by an E3 ligase like cereblon. And we've I feel like the field has gone from being kind of more serendipitous, we found out this happened, to understanding the principles and really designing glues to more of a rational design approach.
Sure.
So what the company kind of like specializes in is essentially using our knowledge base and allowing for recruitment of not something like, BRAF that already has an inhibitor, but things that are like undruggable, like don't have a nice pocket, right? That we can already put a small molecule into. So it's really, our goal is really to expand the druggable space. Yeah.
Thank you for asking your question yourself and giving an answer, because that should have been my question, really. Just asking you to explain what a company does, and you did it brilliantly. So I
felt like I couldn't answer the other question without kind of...
No, that makes perfect sense, and it should be the role of the interviewer, but thanks for being a wonderful guest. and this... that makes sense. So the landscape here is a focus on the undruggable targets, which is fascinating, as is fairly new, I suppose, right? Because usually what pharmaceutical or drug companies do is looking at druggable targets, isn't it?
Yeah, I mean, I feel like going after the undruggables is always someone's your goal, right? You want to go after the difficult to drug things that are are high impact to disease. But I think we have the chemistry essentially to be able to answer these questions in a really impressive and nice way. And it's been really enlightening to work at Monte Rosa. It's been great. Yeah.
There's something I'm interested in. Is you speak with enthusiasm about these mechanisms and this idea. What is it that takes you out of bed in the morning? Is it your sort of fundamental curiosity, so trying to shed light into this mysterious biological mechanisms, or is it the idea that potentially what you do can affect people's life? What is it?
A little bit of both. I feel like the small mechanistic questions you get to answer, I don't know, to me it's super fascinating. That gets me out of bed. And if I have an interesting result that I didn't expect, something is working completely different from what I anticipated, those things wake me up. They get me excited. They get me in the lab. They get me, cranking out results. And also just just expanding the possibility, right? I feel like that's the cool thing of this field in general, being able to say that this wasn't possible yesterday, but today this is a norm. We can't say that that's an undruggable anymore. That's exciting, and not only for the scientific community, but for patients down the line to be able to expand that completely. Yeah. So
you moved from the academic, your academic studies. You don't really have a long-term experience into running an academic research. You moved to industry pretty much immediately. Do you feel? Did you felt a difference in the way things are run? And do you ever feel limited in the way, companies have to run things, because obviously they are for-profit organizations?
I will say no, not at all. I feel like, if anything, I have lots of brilliant people and lots of resources around me to answer a question in a way that in academia it's a little bit more difficult. Sure. The way Monte Rosa is set up, we kind of have excellent people in proteomics, excellent basic biologists, excellent biochemists and biophysicists, and I have them at my fingertips. I have a problem, I shoot them a Teams message, they look at it and they tell me X, Y, and Z. I think it's great. I don't feel limited in that way at all. Yeah.
Can you tell me a bit more about the culture then? Is it, it's obviously not a massive company, so I'm assuming it's flexible and nimble. So does it feel like a university group in some ways, just with more resources maybe and lot of great people?
Where the culture is, it's a pretty relaxed culture. I feel like the senior leadership tries to keep things light and light and kind of like I don't know how to describe it, but we don't take ourselves too seriously, but we're very science driven. We get very excited about the work we do, and we like to do very good work. But we're not I don't know how to describe
it. I understand perfectly what you mean.
Yeah. So I think that, and we're kind of half here and half in Basel. So there's a lot of Zoom meetings and communicating over the computer. But overall, I think we all have a similar personality and a similar feel. Yeah.
It fits you nicely, doesn't it?
Yeah, it fits me nicely. It works.
So what do you think the future, maybe 20 years from now, looks like for Shanique?
Ooh, that's a good question. 20 years from now. 20 years from now, I hope that I have... Oh, that's a good question.
It's a difficult one.
It's a difficult one. There are a couple of ways. There are a couple of things that I feel like where I could be doing in 20 years. I hope to still be in drug discovery. I hope to be leading a great team that is, you know, taking things forward, taking things to the clinic and really pushing the boundaries of what we can drug. I also have like a growing hope to maybe go back to Ghana and also start to establish a research institute there and start to bring a little bit more research to Ghana as well. And I hope to do that within 20 years. So a little bit of two.
Maybe you
very
can
different
do both.
futures.
Yeah,
I hope I can do both. That would be amazing.
You shouldn't forget about this. Sometimes think about it because, you know, you're still young. 20 years is a fairly long time. So yeah, you can make it. And now,
I hope you're right.
Yeah, I'm sure I am. I'm always right. That's what I tell my wife. Anyway, you know, maybe we don't need to wait 20 years, right? You're still very young, but I think you have achieved already quite a lot. You have a good breadth of experience, and you definitely are in a position to actually think about speaking to someone just starting. What would you be your suggestion?
I think the advice I gave there and the advice I'd give now is to remain curious and focus on your excitement about science versus everything else in the world, right? I feel like, or in the world that could be discouraging you about science. So I feel like what works for me or what has worked for me is trying not to think too much about, I don't know, like, oh, I got like an F there, you know, that was terrible. Oh my gosh. But more like, this is the question, it's a scientific question. I'm very excited about this. I'm excited about science. And I feel like paying attention to that versus everything else kind of, it helps you maintain sanity and excitement and keeps you
Makes
going.
sense.
Yeah.
And this is traditionally my last question of my interview, so I'm not allowed to ask you anything else. It was a brilliant way to close it, though. Thank you very much for being with us. It's been good fun speaking to you, Shanique, and I learned a lot. Congratulations for all you have done so far, and best of luck for the future.
Thank you so much.